Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about lactose operon or Lac Operon or lactose metabolism pathway. Okay. In the previous videos we talked about operon concept in prokaryotes. So if you missed this video lecture, so go back to my channel and watch this video because it is very important before understanding the Lac Operon concept. Okay. So let me clean the whiteboard and let's start. Okay friends, so now let me write Lac Operon. Lactose operon, or we also called it Lac Operon or lactose fermented or lactose metabolic pathway or operon. Now what is actually Lac or lactose operon? Again, this is actually the set of genes which contain Lac genes, and these Lac genes are actually responsible to produce those enzymes which are used for the lactose fermented pathway. Okay. It contains promoter region, it contains operator region, it also contains Lac genes as well. Moreover, at upstream, it contains the promoter region and the promoter region and operator region, as well as it contains the Lac inhibitor gene as well. Okay, so this is how the structure. So how we, this lactose operon is actually the part of transcriptional initiation, regulation of transcription initiation part. Okay, and this lactose operon is actually, we also call that this is actually positive transcriptional control pathway. Why? Because this Lac operon, most of the time this Lac operon are switched off. It is not switched on. Normally and naturally this Lac operon is switched off. Okay, but if there is inside the media, if the lactose concentration is higher in concentration, so at that way the lactose will be switched on. Okay, because when it is switched on, so then it will produce some enzymes due to which the lactose is break down. Lactose are taken from the environment and it get back down and converted into glucose as well as in galactose, by which the cell use the glucose portion as an energy source. That's why when the lactose concentration is in high in concentration in a media, so at that time the lactose operon will be means switched on. It will be switched off when the glucose concentration is present. So this is actually the phenomena that how it gets switched on and switched off. Let me explain with the diagram. So what actually going on? Let's suppose this is a structure of the DNA, as you can see. So this is actually the diagram of lactose operon. Okay, this is the Lac Operon structure. So all of you better know this is what this is actually Lac Operon. What? This is Lac Operon. And these are actually the regulatory genes. These are what? These are the regulatory genes which produce either inhibitor enzyme or which produce activator enzyme. So we already talked that inhibitor enzyme can switch off the process of gene expression, and activator enzyme can switch on the process of gene expression. So that's why we call it, these are actually the regulatory genes, and this is actually the operon portion. This is also the operon portion, but this whole is called as Lac Operon portion. Okay, this whole is called as what? Lac Operon. Now why we consider this a Lac Operon? Because it contains all the Lac genes. At that portion, if you see, we have an inhibitor, but this inhibitor is actually Lac inhibitor. And let me write, this is what? This is actually Lac inhibitor. Okay. means it produce the genes or it contain that type of the genes which are transcribed and translated into Lac inhibitor proteins which switched off the lactose operon. Now how? For example, this is actually, as you can see, we have the RNA. As you can see, this is, let's suppose, the mRNA. From this mRNA we get what? We get the inhibitor protein. We get the protein, as you can see. This is what this is, let's suppose, the protein. Okay, and this protein is actually Lac I. What this is? Lac I. So these regulatory genes are responsible for that purpose. That's why this whole is lactose operon. Then we have this, as we already talked, that mostly the promoter region is available for the RNA polymerase. At the promoter region, RNA polymerase are mostly bind at the promoter region. That's why promoter region is also important. There are some sequencing through which the RNA polymerase can bind. Then we have this operon. Now this operon contains the lactose genes. As you can see, this promoter region is important for the RNA polymerase. This operator region is important for the binding. This is actually responsible for the binding of at that portion the Lac inhibitor protein are bind. So when Lac inhibitor protein binds, so then the process is switched on. Then we have another, let's suppose the RNA polymerase is bind here and it will transcribe and produce these three things. So what we can say that this is actually polycystronic gene expression. We also called it Now why we called it polycystronic gene expression? Because in this case we have one promoter which control the structural genes. These are the structural genes and promoter operator are the regulatory genes. So when, remember guys, when we have the different structural genes and it is controlled by one promoter, we called it polycystronic gene expression. So what we get from this, let's suppose RNA polymerase bind here, so it will transcribe and it will produce again mRNA. So we get mRNA, as you can see. But remember guys, this mRNA is called as polycystronic mRNA. Okay, why? Because this mRNA have multiple genes and it produce multiple, it have a genes which produce multiple types of the proteins. So this mRNA which we obtain from this DNA of this Lac Operon, so it will give a rise three types of the protein. One we called it beta galactosidase, let me write beta. Other we call that permease, and fourth we call that thioglycoside transacytylase. Thioglycoside transacytylase, in short form we can write it transacytylase. Okay, now what are the function of these three enzymes? The first enzyme is used to convert the lactose, means let's suppose this is lactose, this is what, as you can see, this is lactose. So it convert the lactose, means it convert the lactose into two-way, because all of you better know the lactose is what? Lactose is actually disaccharide. It is the carbohydrate, means it contains two sugar unit. Why? That's why we call that disaccharide. So the beta galactosidase break down this lactose and convert it into glucose and galactose. Okay, glucose and galactose. So another function of this beta galactosidase is to convert the lactose, means it also convert this lactose into allolactose. So this is actually the function of beta galactosidase enzyme. Then we have another enzyme. This enzyme is especially responsible to make the membrane permeable. So this is responsible to make membrane permeable. So remember guys, if the cell membrane of the bacteria is permeable, so it easily uptake the molecules or the substance which is present outside the cell. So Then let's suppose the lactose which is present outside, so this permease enzyme will make the cell membrane permeable, due to which the lactose is uptake. So this is the job of permease enzyme. It makes the cell membrane permeable. Third, we have transacytylase. Transacytylase have the same function, permeability. But remember guys, still their function is not well explained. Still it is not clear. Sometimes, this transacytylase will break down the this galactose and convert it again into glucose. This is also the function of this transacytylase, but still it is not clear. Okay, so this is all about the enzymes which are produced by this Lac Operon, and these are the main function of the enzymes. Okay, then another question arises in our mind, that what is actually allolactose can do? As the lactose are the carbon source, and the bacterial cell not need this lactose, it need the glucose, which is actually the energy source. Then what is allolactose? Allolactose, again, this is the carbohydrates that convert the inhibitor protein, Lac I protein. It brings some conformational changes in the Lac I protein. So when the conformational changes occur in the Lac I protein, so then what happen? The structure of the protein will be converted, and at that point it will be removed out. What we talked that actually in earlier we talked that most of the time this this portion is switched on, and due to this way this protein is produced, and it will bind to this operator region, as you can see, this operator region. So once it is bind, so then the process switched on. Normally the process switched off, okay, it's not on. But if allolactose we get, so this allolactose bind here, let's suppose this is allolactose, it will bind here, and it will bring some conformational changes, due to which it will removed out from the operator region, and then there are RNA polymerase binded, and it will transcribe these genes and produce these types of the protein. So this is actually allolactose job. So these are actually the main things that are present in Lac Operon. But again, a question arises in our mind, that how all these functioning going on, how this Lac Operon is switched on and switched off. This is the question arises in our mind. Remember guys, inside a media, let's suppose if lactose is present, so then it will cause the switched on, means the Lac Operon will be switched on. If lactose is absent, or let's suppose glucose is present or lactose is not present, let's suppose, so then what happen? The process will be switched off. So this is very important concept. Remember guys, this Lac Operon will be activated at that time when inside the media lactose concentration get high. So at that time the Lac Operon will be switched on. But if the lactose concentration is decreases, or lactose concentration is not present, and we have just glucose concentration, then what happen? This Lac Operon will be switched off. So this is very important phenomenon. You guys need to understand about that. So at that time this is switched on and switched off. Normally what happen? These genes, as you can see, regulatory genes, is switched on, and it produces this mRNA, and we get protein which we call Lac inhibitor, and it bind at that point, which can stop or block the lactose operon, means switched off the lactose operon, and we didn't get these enzymes. Let's suppose inside the media lactose is present. Let's suppose this is let's suppose a media, and inside the media we have a bacterial cell, as you can see. This is, let's suppose, a bacterial cell, and in this media we have concentration of the lactose, this red color representing the lactose. So let's suppose this is a media. Inside the media we have bacterial cell, and this red are the lactose present. So what happened at that time? Inside the bacteria, this beta galactosidase are always present. It doesn't mean that beta galactosidase are produced at that time when it is switched on. It is also present when either this one is switched on or off. It is always present. But when it is present during switched off of the lactose, when it is present, so their function is to convert the lactose into allolactose. Okay, so then what happen? It is converted into allolactose. So when it is converted into allolactose, then what it will do? It will bind here. This is allolactose, bind here, and due to this way some conformational changes occur. Means let's suppose conformational changes will occur. Let's suppose. What kind of conformational changes occur? Conformational changes means the structure of this inhibitor will be converted. As you can see, when the structure is converted, so it will be removed from the operator region. So then it will remove from the operator region. So when it is removed from the operator region, so then this promoter region is free. So what happen? RNA polymerase are bind, and it will transcribe the genes. So this is the process, and we get beta galactosidase, we get the polycystronic mRNA, and then we get beta galactosidase, permease, and transacytylase. This beta galactosidase break down the lactose into glucose and galactose. At that time when we get the beta galactosidase, so then it doesn't mean that they will perform the allolactose function. Now it will convert the lactose into glucose and galactose. Glucose are obtained by the cell as an energy source. This is actually the function. This is actually the switched on of the Lac Operon. How this process will be switched off? This process will be switched, normally it is switched off, but if the concentration of the glucose inside the media are higher, so then what happen? This Lac Operon will be switched, it should be switched off. So then what happen at that point? This means this glucose concentration, let's suppose higher concentration, so that's why it will be switched off. So what happened after that? After that, this gene is activated. Means this glucose, as you can see, this glucose will bind to this portion, to this operator portion, and give signal. Give signal that when it gives signal, so then what happen? We get the mRNA. From this mRNA we get the protein. Okay, so when we get the protein, this is Lac inhibitor protein. It will bind to that portion, to the promoter portion, to the operator portion, and again switched off the process. Why? Because just consider, if we have already glucose, then why this process is switched on? Okay, the process will be switched on when we have lactose, because this gene is specially responsible for the lactose, breakdown of the lactose. If the lactose is not present, so then why we get these? Then these enzymes are not needed. These enzymes are not needed. If the lactose is present, these enzymes will be needed. Okay, so this is how the Lac Operon is turned on and turned off, switched on or switched off. Okay, but still there is some important things that are present in the lactose operon system that are also responsible for the switching on and switching off the lactose. We call that cap and camp. Okay, these two are also some molecules or proteins which are actually responsible for the switch on and switch off the lactose operon as well. Because we also talked that how it is switched on and switched off, but these two are also responsible for the switching on and switching off the lactose operon respectively. Now remember, if glucose is present, let's suppose, and lactose is absent, let's suppose, then what happened? This is the process. If let's suppose glucose, glucose is present and lactose is present, then what happened? Another question, if let's suppose lactose and glucose is absent, both are absent, then what happen? These are the questions arise in our mind. We just talked that if glucose is present, the process will be switched off. Lac Operon will be switched off. If the lactose is present, the Lac Operon will be switched on. We just talked that. But we didn't talk that if glucose and lactose is present, what happen? If glucose is present and lactose is present, what happen? We didn't talk that. So what actually this cap and camp means? Cap is actually the proteins. These are what? These are actually the proteins. And camp is actually small molecules or effector molecules. We call that effectors molecule that act upon the cap protein, or we can say that it will help to activate the cap protein. We can also say that. Now what happened to explain that phenomena? Again, let's suppose we have what we have the this is, let's suppose, again a DNA, as you can see. So let's suppose this is the DNA structure. Now to explain, let's suppose this is the DNA structure. What happened? If the glucose concentration is present, so then we have and lactose is absent, so then what happened? Camp means camp concentration get decreases. Okay, if camp concentration is decreases, so then what happen? Normally what happen? We have already, cap protein. This is, let's suppose what, this is actually the cap protein, as you can see. This is actually cap protein. And this is camp, as you can see. This is what, this is camp. So when cap and camp connected to each other, so this enzyme is converted into holoenzyme. This is actually appo enzyme, means inactive form of the enzyme. So when it is connected, so then it is converted into active form of the enzyme, which we call it holoenzyme, and we get a complex. This complex is called a cap-camp complex. It will bind to this cap binding site. So when it is bind to this cap binding site, so what happen? It bring RNA polymerase. It bring RNA polymerase. RNA polymerase will sit here. After that, what happen? RNA polymerase will sit here, and it will transcribing the genes. So it will sit here at the promoter region and transcribing the genes. So this is also the switched on process of the Lac Operon, that how it is actually switched on. Okay, so it will be switched on when the glucose concentration is present, so the cap concentration get decreases. If let's suppose glucose concentration is if the glucose concentration is present, cap concentration get decreases. If lactose is absent, again the cap concentration is decreases. So remember, so when the cap concentration is decreases, so we do not have the camp. If the camp is not present, it will not bind to this cap protein. If it is not bound to the cap protein, it will inactive form. And if it is in inactive form, so again it is not bind to this cap binding site, due to which RNA polymerase will be not binded and the process will be switched off. This is how through this cap-camp complex the process is switched off. What happened now? Sometime, if the glucose concentration is absent and lactose concentration, let's suppose, if it is absent and lactose concentration is present, then what happened? At that time we have again the cap, but at that time the cap concentration will be increased. So if the cap concentration is increased, it will bind, cap protein bind, we get cap complex, and it will bind to this cap binding site. Then RNA polymerase will be attracted, and the transcription of this genes will be going on, and we get polycystronic mRNA, and after that we get all these protein. So this is how through this complex the process is switched on. Okay, now remember, if both glucose and lactose is present, what happen? The process is again switched off. Why the process is switched off? Because we have glucose, the cell using the glucose, the cell didn't use the lactose. The process is switched off because glucose is present. Lactose is also present. If the glucose is used by the cell in a large number, then you know what happened? This lactose concentration is increasing because cell using glucose again and again, and if the glucose concentration get decreased and lactose is increase, so again the same process will happen. If let's suppose so we have this two, means if we have, let's suppose if we have lactose, not lactose, we have not glucose, again this process is switched off. Why? Because lactose is not present, then why this Lac Operon will be on? Lac Operon is on at that time when the lactose concentration is higher. If lactose is not present, why it is on? If glucose is not present, then why it is on? This is the phenomenon. So if glucose and lactose is present, it is switched off. If lactose and glucose is present, it is again switched off. It will be switched on when the glucose and lactose, if anyone is present or absent, so then it will be switched on and off. But if both are present and both are absent, it will be switched off at a time. Okay? But if any one of them are present or absent, it will be switched on or switched off. So that's all about the lactose operon and how it is switched on and switched off by the glucose and lactose. And we also talked that how it is switched on and switched off by this cap-camp complex. Okay, so I hope you guys get idea about the lactose operon. If still you have any kind of question, you can write your question through a comment. Inshallah, I will try my best to give you answer as soon as possible. If you like this video, so make sure to hit the like button, share this video, and definitely subscribe my channel to get more interesting videos like that. Thank you so much for watching.